Hello बच्चों, let's see the question. So the question says restriction enzymes are. So इन चारों options में से हमें identify करना है the best statements which describe restriction enzymes correctly. So the key concept in this question is to understand what are restriction enzymes. So this is an easy question, and let's understand what is a restriction enzyme. So first of all, restriction enzymes are also known as restriction endonucleases. So restriction endonucleases are specific types of nucleases that can cleave DNA at a particular site or at a specific site only. So every restriction endonuclease recognizes a sequence of nucleic acids which is known as the recognition site. So different restriction endonucleases have different specific recognition sites. So once the restriction enzymes recognizes these recognition site, it cleaves the DNA into two fragments by cleavage at a specific site, and this site may be either present inside the recognition site or at some other place other than the recognition site. So the cleavage of the DNA by restriction endonuclease is very specific. Now this enzyme is particularly found in bacteria. So this was first discovered in bacteria because this enzyme is a protective mechanism for bacteria against bacteriophages. So the restriction endonucleases present in bacteria are specific for the viral DNA. They recognize the viral DNA and they cleave the DNA into two fragments, resulting in making the viral gene non-functional. And therefore, the bacteriophage cannot cause an infection in the bacteria. So it provides a protective mechanism in bacteria. Now, the discovery of this enzyme was very useful for recombinant DNA technology or genetic engineering. So, since restriction endonucleases are site-specific nucleases, they can be used in recombinant DNA technology for the insertion or deletion of a specific sequence of nucleotides. So the discovery of this enzyme helped us in making recombinants like recombinant DNA or plasmids for cloning of a gene of our interest into different organisms. So restriction endonucleases play an important role in genetic engineering. So if we come back to the options given, so the first option says that not always required in genetic engineering. So restriction enzyme plays a very important role in genetic engineering. So this won't be the correct option. So the second option says that it is an essential tool in genetic engineering. So this is the correct option because we just now saw that recombinant DNA technology wouldn't have been possible without the restriction enzymes. The third option says that it is a nuclease that cleave DNA at specific sites. So again, this is the correct option because restriction enzymes are restriction endonucleases that cleave DNA at a specific site. So we can see that both option 2 and 3 are correct. Therefore, our final answer for this question will be option number 4. That is 2 and 3 both are correct descriptions of restriction enzymes. So our final answer is option number 4, that is 2 and 3 both. Hope you understood this well. Best of luck. Thank you.